But I'd like to introduce our keynote speaker. It's a good friend who I've known for almost 20 years, at least 15 years at Seattle Children's Hospital. Troy Torgerson is the consummate human immunologist who I've gotten to know over the years. He did his MD PhD at Vanderbilt and then came out and did a pediatrics residency at Seattle Children's, University of Washington. Quickly realized what he wanted to do and crafted his own fellowship, a combined rheumatology, immunology fellowship, and worked with a variety of people in rheumatology as well as Dr. Hans Ochs in his immunology lab. He made fundamental discoveries in the field of IPEX syndrome, which is immune dysregulation, polyendocrinopathy, enteropathy on the X chromosome. Subsequently has worked on Wiskott-Aldrich syndrome, hyper-IgE syndrome, as well as a variety of other collaborations with other people looking at various immune deficiencies. And what's really come out over the years is that Troy is one of our connectors. He connects people who work in the primary immune deficiency field with people who are thinking about therapies. He set up a very good, tight relationship between our non-malignant transplanters at the Fred Hutchinson Cancer Center with our group and other non-malignant hematology providers. He's gone on to become co-director and then director of the Immunology Diagnostic Laboratory at Seattle Children's Hospital, and is one of only the few places in the country that is really looking in great clinical detail for our primary immune deficiency disorders, and is a great resource for everyone across the country. I don't have too much more to say. He's been very active, and what I enjoy most about him is his ability to make the connections, both on a clinical basis and on a research basis.
Thanks, Mark. That's an incredibly generous introduction. Thank you.
Okay.
I told Mark that I was only coming here so I could recruit him to come back. So I know, I'm just teasing. Our nurse did, however, though, said, please bring Mark back with you. So anyway, I know you guys have found how valuable he is here. So your gain is our loss for sure. So I wanted to talk today about STAT defects. So this family of transcription factors we've known about for now about 23 years, somewhere in that neighborhood. And despite all that we know about these transcription factors, we're actually learning new things about them all the time. And they've become incredibly important actually in human disease. And the really fascinating thing about these transcription factors is that as I'll show you, depending on what the defect is, they can either have, the defects can either decrease STAT function or they can increase STAT function. And depending on that particular defect, it actually dictates the clinical phenotype, which I hope will become clear as I walk through this. My disclosure site, so I accidentally left it out. I apologize to the CME folks, but there's no disclosures relevant to this talk. So anyway, so the topics that I want to cover today are just, for those of you who don't do stat biology all the time, just a reminder or review of stat biology. Then I want to sort of walk through the stats that have now been associated with human disease. So STAT1, loss of function mutations and gain of function mutations. STAT2 defects, these are actually loss of function mutations. STAT3 defects, loss of function and gain of function. STAT4 defects, STAT5B defects, and then I'm going to talk a little bit about mechanisms, a little bit of the work that we've done around particularly the STAT3 loss of function mutations, which the patient mutations have really directed us in that work. So just as a reminder that this is a family of transcription factors. There are 7 in total of STAT family members. And just to remind you how these work, so the STAT proteins are actually present in the cytoplasm as non-phosphorylated dimers. So we used to think that they were monomers. Now we know that they actually exist in dimers, and they bind together in this sort of a head-to-tail fashion with the SH2 domains sort of far away from one another. And I'll come back and describe the domain structure in just a moment. When cytokine binds to its receptors, and there are a number of cytokines that act through the JAK-STAT pathways, but when cytokines bind to their receptors, they aggregate these receptors, and oftentimes these receptors are associated with these JAK kinases, of which there are 4 family members. And these kinases are very tightly bound to these receptors. The affinity is so high that some people even consider them almost as a single unit because the affinity between the JAK kinases and the receptors is so high. The cytokine binds, it aggregates the receptors, causes the JAK kinases to phosphorylate the cytoplasmic tail of the receptors on specific tyrosine residues. The STAT proteins then use their SH2 domains to bind to the phosphorylated tyrosine residues, allowing them to be phosphorylated on a specific tyrosine residue. In each case, they're phosphorylated on a specific tyrosine residue that causes then this dimer to undergo this huge conformational change and flip from sort of a head-to-tail conformation to a head-to-head conformation where the SH2 domains of each other, they bind to one another's phosphotyrosine residue. And I'll show you that in just a second. Once they do that, they then transit to the nucleus where they bind DNA. Now, one of the things that we know, based on a lot of work done primarily on STAT1, is that actually the STAT proteins are cycling in and out of the nucleus constitutively. They circulate into and out of the nucleus, and it's when they get into this, into the specific conformation that they need to be into to bind DNA, what happens is they bind to DNA, and that masks the nuclear export signal so that they're then stuck on DNA until they can be dephosphorylated and open up that export signal and be exported back out to the cytoplasm. Okay, so that's sort of a brief reminder of STAT activation in biology. Now, what's interesting is that despite all that we know about this protein family, there's still a lot of questions that are really not known. We don't know really how this big conformational change in the dimer happens. We don't know how the STAT proteins actually get ejected off of the receptors. It was thought that maybe it had to do with the phosphorylation of the tyrosine on the STAT molecule, but that's not so clear actually. And it's not totally clear how they get downregulated once they're in the nucleus, although it's known that there are phosphatases that are required to dephosphorylate the STAT proteins. Okay, so just as a reminder of the domain structure of the STAT proteins, we're going to sort of ignore the JAK proteins for the rest of this talk mainly, except for the end when I'll come back to them briefly. But to remind you of the domain structure of the STAT proteins, there's an N-terminal domain, a coiled coil domain, and in the center is a DNA binding domain. There's a domain that's called a linker domain, then the SH2 domain, and then a domain that we call the spacer loop, and then that provides a little spacer between the SH2 domain and the critical phosphotyrosine residue, and then the transactivation domain. And what this looks like, of course, is here. The crystal structure for several of these has been solved, and so this is the STAT3 dimer bound to DNA. This is the STAT1 dimer bound to DNA. Looking down the barrel of the DNA here and here, the structure is very similar. The N domain is gone here, so you've got the coiled coil domain in purple. Same here, coiled coil domain. DNA binding domain in red. The linker domain here. The SH2 domains up here, and so you can see how the SH2 domains, they have that little spacer, and then the critical phosphotyrosine residue is here, and they put that in the pocket of each other so that this sort of pins them together, almost like a pair of pliers, so that they can bind to DNA. And then, of course, the transactivation domains sit up here, and draw in the basal transcription machinery to induce transcription at specific loci in the genome. So the structure of these is all quite similar, and this becomes important as we go through this talk, because I'll be referring to the structure sort of repeatedly because it does affect the phenotype actually of the patients. So one of the things that I'm going to be talking about as I go through this talk is the fact that there have now been loss of function mutations identified and gain of function mutations. And many of these mutations are heterozygous. So these are autosomal dominant defects, many of them, meaning that you have two alleles, for instance, that encode STAT1. You only need one mutation in one of those alleles to generate disease. And so you've got a mixture in the patients of 50% wild type protein, 50% mutant protein, okay? And so stochastically, if you think about how this all shakes out, you're going to have about 25% you'd expect to be wild type dimers. You're going to have 25% that are mutant wild type, 25% that are wild type mutant, and then 25% that are mutant, okay? So if you get one of these point mutations that either downregulates STAT function or upregulates it, it's going to affect essentially 75% of the pool of STAT proteins, okay? And so that's how this works. Now, to give you an example of why this is important as we think about these diseases, so let's talk about STAT3. So I'm going to talk in a few minutes about STAT3 loss of function mutations that have been identified in patients with hyper-IgE syndrome. And with STAT3, we know that complete knockout of STAT3, at least in the mice, and we predict in the humans as well, complete knockout is an embryonic lethal, okay? Now, in the mice, if you have a knockout of one allele, so you just completely knock out one allele, those mice have no discernible phenotype. They seem completely normal. And interestingly, we have never identified, even though, as I'll show you, we've identified many humans with mutations in STAT3, we have never identified a patient who has a mutation that completely knocks out the function of one allele of STAT3. There's no deletion. It's frame shift mutations. There's no complete deletions of the gene. All of the mutations found are in-frame deletions or point mutations, meaning that that protein, the mutant protein, has to be expressed in order to downregulate that other allele. So 50%, we think, is 50% STAT3 function is enough to survive and have no disease, but 25% is enough to be born, but not enough to keep you disease-free. So it's interesting, and it brings up some really interesting questions about the threshold and sort of the redundancy of these pathways in preventing human disease, okay? So that's an important concept, I think, to get before we move on through these diseases, okay? So with that, so the status of things now. So there have been loss of function and gain of function mutations identified in humans in STAT1. STAT2, there's been loss of function mutations. STAT3, loss of function mutations. I'll show you a tiny bit of data about a new defect that's just been identified, a loss of function mutation in STAT4, loss of function mutations in STAT5B, okay? Now, these others have not yet been identified, but given that we've found all these others, I would predict that many of these green dots will eventually be filled in human diseases, okay? So we'll walk through these one by one. So let's start with STAT1 defects. And first, I'll just remind you that STAT1 is the major signaling pathway, particularly for the interferons. So gamma interferon and the the type 1 interferons, alpha, beta, et cetera, that act through pathways, receptors that ultimately activate STAT1. In the case of the type 1 interferons, they also activate STAT2, which will become important in the next set of diseases that I'll talk about in a moment. But we're familiar with the role that the interferons play in preventing infectious diseases. And so it turns out that these patients have infectious disease problems, the patients who lose STAT1 function. So let's talk first about STAT1 loss of function mutation. So These are some of the mutations that have been identified. This is from a review article published by Jean-Laurent Casanova, whose group has been absolutely involved in all the identification of all of these defects. And in brown here, right over here, so in brown, these are the severe mutations. Many of these mutations are autosomal recessive. Actually, the patients have mutations in both alleles and lose complete STAT1 function. And these patients have a severe phenotype that is the hallmarks of that phenotype are that they get invasive mycobacterial infections with non-pathogenic mycobacteria, and they get severe viral infections, as you would predict, not being able to have either interferon gamma or type 1 interferon signaling, okay? And these patients, many of them die fairly young because of this defect. There's also an autosomal recessive form that has milder mutations that are in blue. These are point mutations here in blue. And then an autosomal dominant defect, where again you only need one mutation in one allele, acts in an autosomal dominant fashion, dominant negative way, that's also a milder defect, that those are shown in green. And these two forms of milder mutations tend to have the mycobacterial susceptibility, but much less of the viral susceptibility that is seen in the severe forms. So mycobacteria is the big player in these patients, and viruses in the patients who've got a complete knockout. And this is terrible disease if they've got it. We've got a patient who's in our hospital right now who has an autosomal dominant loss of function mutation in STAT1. She is absolutely chock full of Mycobacterium avium intracellulare. And it's very difficult to eradicate. The organism is very resistant, and so she's on interferon gamma supplementation to try and drive that 25% of normal STAT1 complexes that she has, but it's not moving very quickly. And she's still alive, but we're not sure if she's going to make it, actually. So those are the STAT1 loss of function mutations. Interestingly, the STAT1 gain of function mutations have a completely different phenotype in general. These patients were originally described by Jean-Laurent's group and by another group in Germany as having chronic mucocutaneous candidiasis. And the original description was that these patients had almost exclusively CMC and not much else. There were a few of them that had susceptibility to cutaneous staph infections, but CMC was the overwhelming disorder that these patients have. Now, there are a few of these patients that have now been described who also have some susceptibility to fungal infections and certain mycobacterial infections, and this is what we originally thought this defect looked like. And then and this is the chronic mucocutaneous candidiasis that some of these patients have. Some of these patients also have aneurysms because of the role that STAT1 we think plays in some of the vascular genesis and other things. But the gain of function looks like this. So this is one of our patients, and so this is in green is cells from the patient that are not stimulated. In this case, we've stimulated the PBMCs either with IL-6 or IL-27. And in blue is a normal individual. And looking again at early time points at STAT1 phosphorylation by flow, 7.5, 15, and 30 minutes. And you see the normal individual phosphorylates STAT1, it goes up, and then it sort of comes back down as with the induction of phosphatase activity in the SOX proteins. But it doesn't return, in a normal individual, doesn't return to baseline. In red is a patient with one of these STAT1 gain of function mutations. They're hyperphosphorylated, and they stay hyperphosphorylated for a long time. And so the cells presumably feel like they're under this just massive cytokine drive, and their gene transcription will mirror that. But in reality, this just requires normal doses of the cytokines. So this is what the gain of function looks like. And we noted, we had actually shared some patients with Steve Holland at the NIH who had an IPEX-like phenotype, so autoimmune disease with severe autoimmune enteropathy. Many of them had other features. And Two of the patients that we had shared with him had also chronic mucocutaneous candidiasis, and so we thought maybe they could have this disorder, and we sequenced, and sure enough, that was the case. So altogether, we identified initially in the initial cohort that we published 5 patients, and let's see, 5 here. We had 6. The 6th one we didn't have the data, so it didn't make it into the paper. But these patients had enteropathy. They had endocrinopathy, so diabetes, thyroid disease, much like the IPEX patients. They had skin manifestations, so many of them had eczema, and then other forms of autoimmunity, but they also had chronic mucocutaneous candidiasis. And that was the clue. And these patients had STAT1 gain-of-function mutations. We've now found a total, in our cohort of IPEX-like patients, we've found a total of about 10 patients who have this phenotype. It is curable by bone marrow transplant. Some of these patients have been transplanted and have done well, but the mechanism by which these patients develop autoimmunity is not entirely clear. They have, it appears that regulatory T cell function in the few patients we've been able to test it seems to be mostly normal, but we think that it's maybe the target cells that can't be downregulated as easily because of this massive drive of STAT1 that they have. That's completely hypothesis. We don't yet have the data to prove that. Hopefully we'll have that soon. So these are the STAT1 gain-of-function mutations, and there are a lot of these patients out there, actually. There's quite a number of these patients, and the mucocutaneous candidiasis can be quite mild. Some of the patients only get it when they're on antibiotics for a while, and then they have to be put on antifungals each time they're put on antibiotics. That's STAT1. STAT2, again, remember that STAT2 is critically involved in responses to type 1 interferons, and so you can sort of predict what a loss of function mutation in STAT2 may do. And in fact, this is recently published data from Sophie Hambleton's group. They found a couple of patients in one family who had a transposable element plugged into the STAT2 gene such that it was created a truncated protein, and so loss of function of STAT2, and these patients have, as you might predict, disseminated viral infections. And interestingly, what was most problematic in both patients was vaccine strain measles. And these patients became very ill. One of the patients actually died, and of a fatal viral infection. So, as you can predict, STAT2 defects, loss of function mutations, have a phenotype of viral susceptibility. And again, the hunt is on for more of these patients, but to my knowledge, these have not yet been found. It's not clear to me what a STAT1 or STAT2 gain of function might look like, but so we may or may not find those, but you might sort of hypothesize what those patients might look like. Now let's talk about STAT3 defects. And STAT3, as you know, plays critical roles in many, many cytokine pathways. Important cytokine pathways including IL-6, LIF, IL-11, IL-10, IL-6, IL-22, 23, there's a whole host, IL-21 that signal through STAT3. So STAT3 plays a critical role, which is why patients, that's supposed to say STAT3 loss of function, sorry about that. Patients with STAT3 loss of function defects have a systemic phenotype. So these patients were originally actually described in Seattle by the person I consider my scientific grandfather, Ralph Wedgwood, who trained Hans Ochs, who trained me. And Ralph and colleagues described these 2 red-headed girls who had this phenotype of cold abscesses. So they'd get abscesses, they'd be large, completely full of pus, but very non-inflammatory. These girls also had, sort of as they matured, development of coarse facies. They had they had bones that broke easily. They didn't lose their primary teeth. All sorts of problems, and they were both red-headed, which initially, actually for a while, was thought to be part of this disease, and it's really not. But anyway, so this is hyper-IgE syndrome or Job syndrome, and actually this is this is probably one of the last times the Bible was quoted in the Lancet. But anyway, it's there. So went Satan forth from the presence of the Lord and smote Job with sore boils from the sole of his foot to his crown. Anyway, so that's how these patients had. They get these non-inflammatory boils. So these patients have bad eczema. They get these cold abscesses, skin and subcutaneous abscesses. They have real problems with staph infections. They get lung abscesses, and they form pneumatoceles, which creates a lot of problems and ends up being the thing that causes much of the morbidity and the mortality in these patients. And these are caused typically by staph. They have, again, in addition to staph, they have fungal infections, bad candidal infections, usually cutaneous, but can be invasive. Coarse facial features, as I showed there. They get scoliosis, fractures, non-traumatic fractures. They're hyperextensible. They retain their primary teeth, don't lose those, and they get aneurysms that have been described both in coronary and brain. And just to show you visually what this looks like, again, this is one of the two girls as she matured. She died shortly after this. You see the fungal nail infections, a pneumatocele here that's actually super infected with a fungus, the hyperextensibility, the scoliosis, the aneurysms, the teeth that don't fall out, and the eczema. Okay, so that's this disease. And from a laboratory standpoint, they've got high IgE, they have eosinophilia, they have positive allergy tests, but they're not very atopic. So interestingly, they don't have severe asthma. They don't seem to have a lot of food allergies, interestingly. There's a chemotactic defect that's been described in the neutrophils, abnormal antibody responses, and B-cell maturation, and abnormal cytokine production. And again, it's caused by loss of function mutations in STAT3, which is involved in all of these pathways. Now, I'm going to come back to this part in more detail at the end and tell you about some of the sort of biochemical work we've done to try and characterize these mutations and learn more about stat biology based on those. So let me move on, and then we'll come back to that. So recently, there were also patients described with STAT3 gain of function mutations. So these, again, are just point mutations. So if you note here, these are point mutations in the gene. And these mutations upregulate STAT3 function. Now, interestingly, these STAT3 gain of function mutations were originally described in certain lymphomas as being present in the lymphomas and potentially driving the disease. What's interesting, so LGL was the lymphoma most commonly associated with these STAT3 gain of function mutations. So it was a little bit of a surprise when they found germline STAT3 gain of function mutations, and the patients didn't all have malignancies. So that was a little bit surprising. And so here are the first 5 patients that were described. So in this paper in Nature Genetics, they sort of compared the phenotypes of what the activating STAT3 mutations look like compared to what the loss of function mutations look like that I just described with a high IgE and eosinophilia, et cetera. These patients who have gain of function mutations instead have autoimmune disease, severe autoimmune disease with severe early onset diabetes. 4 out of the 5 patients had that. Autoimmune enteropathy, autoimmune hematologic disease. Some of the patients, many of the patients, 100% had short stature, and I'll show you that in a minute. They had eczema. Some of them had dental anomalies. So the phenotype looks a little bit like patients with IPEX, with enteropathy, diabetes, and skin disease. There was a subsequent paper published with 13 more patients that were collected in England, the NIH, and in St. Louis. And these, again, have some of the same mutations described in the other paper, and these were the clinical phenotypes of the patients. So note here that many of the patients had, not all of them, but many of them had endocrine problems that were diabetes or thyroid disease. Enteropathy was common in this disease, in this group, and then other autoimmunity. Many of these are hematologic autoimmune diseases. And the thing that we've come to understand about these patients is that the growth phenotype is really profound. And so I apologize for some of you, I showed this to you yesterday, but this is a little girl, we recently identified the mutation. We'd actually been looking for the mutation in this little girl for a few years. She presented with a classic IPEX phenotype. She looked, severe early onset autoimmunity, bad enteropathy, diabetes, and but then she had this tremendous growth phenotype. So ultimately, she underwent bone marrow transplant, and she's now several years post bone marrow transplant. This is her at age 11 with her 16-year-old sister. To give you the idea, to give you a sense of this growth phenotype, and it is really dramatic. And they're not all this small, but many of them have. They all have growth abnormalities. So this is really interesting. And in the paper published by Josh Milner from the NIH, he looked at phosphorylation of STAT5. Of course, STAT5 lies downstream of the growth hormone receptor, and showed that there's been this long-standing, we know that STAT3 and STAT5 have antagonistic effects on many promoters. And so when STAT3 is activated, it turns down STAT5 activity and vice versa. And so you can imagine in a case where you've got chronically activated STAT3, you might see decreased STAT5, and in fact, that's the case. So here is, in red, are patients with these with the mutations, these activating mutations in STAT3. So non-activated cells are shaded. Red are patients that have been activated with cytokine, and looking at STAT5 activation here. And this red are patients, blue are normal controls in each case. And as you can see, the patients all have sort of decreased STAT5 activation, which again, we think is caused by this chronic STAT3 activation that drives this decreased STAT5 activation. And so we're not certain, but we think that this may play a role in the dramatic growth phenotype that these patients have. And I'll show you the STAT5 patients in just a few minutes and what their growth looks like as well. But we thought that this little girl actually had STAT5B deficiency for a long time. I actually made the folks in my lab sequence it a couple of times just to be sure before we found this defect, okay? So those are the two STAT3 defects. Again, on the one side, you've got loss of function, which gives you one phenotype. On the other side, you have gain of function. And what's interesting is that the loss of function mutations, the hyper IgE syndrome patients, autoimmunity is quite uncommon in that disease. Whereas on this side of the spectrum, when you rev up STAT3, autoimmunity is very, very common, and they don't have the infectious phenotype. Really interesting in terms of thinking about just hitting the sweet spot with STAT signaling. So let me just introduce STAT4 signaling. Remember that STAT4 lies downstream of the IL-12 receptor and is critical, of course, for driving the production of interferon gamma by T cells that feeds back on the myeloid lineage cells to help them deal with organisms such as mycobacteria and fungi. And so we know that patients who have defects in IL-12 or in the TYK2 kinase that lies downstream of the IL-12 receptor, so IL-12 receptor, and TYK2 kinase, these patients are among those that have problems with invasive mycobacterial infections. So recently, one of our collaborators in Brazil identified a patient, actually a father and son, who had invasive paracoccidiomycosis infections. And on sequencing, the patients were found to have a heterozygous mutation in STAT4 in the SH2 domain. And this is looking at STAT4 phosphorylation in response to IL-12. So here's a normal individual, non-stimulated in the dotted line, stimulated in gray. Here's the patient, doesn't shift very much, and the dad doesn't shift very much. And I think this is the mom over here, and she shifts normally. So we're in the process of doing the functional work to demonstrate the precise location at which this mutation blocks STAT4 function, but this work will be presented at the CIS meeting in a couple of weeks by Lena Shimke in Antonio Candino's lab from Sao Paulo. And so this is coming down the line. Of course, the hunt is on for more patients with this defect. But again, the phenotype here is invasive fungal infections, as you might predict, again, from not having function of STAT4. Lastly, let me talk briefly about STAT5B defects. So just to remind you that STAT5, there are two subunits. There's a 5B subunit and a 5A subunit. They play critical roles in response to a whole bunch of cytokines, but particularly important for T cells are interleukin-2, interleukin-7. For NK cells, interleukin-15 is important. And so there have now been patients described with mutations in STAT5B. Most of these mutations are autosomal recessive. There is one that's autosomal dominant, but these are all loss of function mutations. And so you can kind of predict what effect these patients might have losing STAT5 function. And that is that they've got a phenotype that's sort of SCID-like a little bit, but it's also got a flavor of IPEX. So this is sort of a mix between SCID and IPEX. These patients I'll get back to the dwarfism in a minute, but they've got some phases. They've got an immune deficiency with T cell lymphopenia and NK cell lymphopenia, and as a result, they get recurrent viral infections. Related to this, they have immune dysregulation, and they get severe enteropathy. They get early onset eczema, and they get an interstitial lung disease that's actually very difficult to control and is the thing that actually causes many of these patients to die, actually. These patients, this is work done by Kari Nadau's lab, showing that in patients with STAT5B deficiency, they have very few FOXP3 positive Tregs. Now, this is the reason for this is that when FOXP3 gets expressed initially in T cells as they're being generated in the thymus, it requires ongoing IL-2 stimulation to prop up FOXP3 stimulation, or to maintain FOXP3 stimulation over the lifespan of the regulatory T cell. And so you can imagine, in the absence of IL-2 signaling, because STAT5 is gone, it can't maintain this signal to drive FOXP3 expression, and so they have very few FOXP3 expressing Tregs, which we think is, of course, part of what drives this autoimmunity. Their growth phenotype is profound because, again, growth hormone signals through STAT5B. And so these patients have dwarfism, as you can see here. Here's the normal growth curve. Here's their height, and it's profound growth failure here in these patients. So that's the phenotype. These are the patients that have been described so far in the literature, and the mutations that have been described. As you can see, they sort of span through the different domains of STAT5. So we have now sort of reviewed some of the we've reviewed stat biology a little bit. We've talked about the defects in all of these different proteins. And now I wanted to just talk briefly about some mechanisms. And mechanisms that we've worked on as a result of the patients with STAT3 loss of function mutations. Now, the reason that we focused on these is because we had this whole cohort of patients with hyper IgE syndrome. We had access to a lot of mutations, and the mutations clustered in different domains of the protein, as I'll show you in just a minute. And STAT3 has become incredibly important from the standpoint of cancer. It is constitutively activated in many cancers, and in most carcinomas, actually, it's activated. And so our thought was that if we could understand maybe a little bit better how these dominant negative mutations downregulate STAT function at the different phases, that might begin to provide us some insights that could be used for potentially therapeutically down the line. So this was the mutation in one of those first families that I showed you, one of the first two girls that was still described, that were originally described. The girl on the top of that picture, she died at age 17. We still don't know her mutation, but the one lower was still alive when the mutations were identified in STAT3. And so she was the first. She and actually her son, her grandson was in our ICU with a pneumatocele from his staph pneumonia when we identified the gene defect in this disease. And so we were beat to the punch by the Japanese, and then in this family, we published this in the same issue of the New England Journal with Steve Holland's paper. So there were several groups that got to this at the same time. This is our cohort, and there's an NIH score that can be used to sort of score these patients, and so these patients had a variety of different mutations that allowed us to sort of dig into this a little bit more deeply. So here are the mutations that the patients have. So the mutations cluster in the DNA binding domain. They cluster in the SH2 domain. We have one patient who's got one family with an interesting mutation in this linker domain that I'll come back to in a couple of minutes. There are a small number of patients who have mutations in the critical phosphotyrosine residue, the Y705 residue, and there are a number of patients with mutations in the transactivation domain. So again, just to remind you of the domain structure, looking at it here, coil domain, DNA binding domain, linker domain, SH2 domain, and transactivation domain up here. So we'll come back to that. Okay, so here are the patients. So we've got patients with these mutations in different domains, and so what we've done is basically gone after trying to understand how they downregulate STAT3 function. And we've done this, we've tried to do it in a systematic way. We've looked at dimerization, we've looked at binding to the phosphotyrosine residues on the receptor, we've looked at phosphorylation of the STAT proteins themselves, we've looked at nuclear localization, we've looked at DNA binding, we've looked at gene transcription. And in looking at the nuclear localization, it's actually allowed us to also look at this phenomenon which is described in several signaling pathways, which is the formation of the signalosome, which is basically a vesicle that's budded off the membrane when cells are stimulated with cytokine, and it moves toward the nucleus, continuing to signal the whole time, and then it dumps off the STAT proteins just outside the nucleus, and then they move into the nucleus where they bind to DNA, okay? So I'm going to just show you one example of this. This is a ton of data to go through, and I won't show you it all, but I'll show you a summary in just a second and then come back to two other interesting ones that we've found interesting. So just to show you, for instance, phosphorylation and the difference based on the domain where the mutation is. And you might predict some of these. So here's a normal individual stimulated with a fairly broad dose response of IL-6. And so here's a normal individual. They have some of the cells respond to IL-6 and are phosphorylated normally. Patients with the DNA binding domain mutations, as you might predict, they're far away from the phosphorylation site in the SH2 domain. They're phosphorylated normally, whereas patients with SH2 domain mutations are not phosphorylated normally. There's a dramatic decrease in phosphorylation. Similarly, the patients who have let's see here. This is a patient with a mutation in that the spacer loop domain, and then a patient with a mutation in the transactivation domain here. So again, these are down around the end where the phosphotyrosine residue is and where the SH2 domain are, and so these don't get phosphorylated normally. And this, of course, in the SH2 domain mutations, we know that that's because it's never able to actually dock on the receptor, and so it doesn't get phosphorylated. The story is more complex here. But in summary of these, this is the story. So the bottom line is that all of these mutations lead to decreased gene transcription, but they block up at different stages. And so interestingly, many of these, almost all of them, have abnormal nuclear localization. The DNA binding domain mutants, again, because the STATs cycle in and cycle out, if they can't bind to DNA, they just get cycled right back out. And so many of them have nuclear localization abnormalities. Many of them, some of them don't bind to DNA, and some of them are not able to bind to the phosphorylated receptor. And so this has allowed us to really delve into STAT3, the basic biology of STAT3, and there's a couple of surprising things that came out of these. One of those that was really surprising to us was that patients who had mutations in the critical phosphotyrosine residue. They get activated, the protein goes to the receptor, it gets bound on the receptor just fine, but interestingly, it can't get off the receptor. It's stuck there, and the cells are not able to initiate formation of the signalosome. So it tells us that phosphorylation on that critical tyrosine residue triggers the formation of the signalosome. And we've got some additional work going on to try and understand how that might work. We don't understand it yet, actually. And again, it's providing it's fantastic that the patient mutations, these experiments of nature, are providing us with insights into the basic biology of the STAT proteins. Let me show you a couple that are particularly interesting, that have provided us some insights into a couple of areas where there's not a lot of data. One of those is this mutation, which I mentioned is this patients who have a 10 amino acid in-frame deletion in this what we call the spacer loop. So just again to show you, here's the crystal structure, the SH2 domains, and we're going to focus in on the SH2 domain. So this is looking down on the crystal structure from the top, and I've just drawn on top of that. Here's one SH2 domain, here's the other SH2 domain, and here's what I'm talking about, the spacer loop. So there's this loop of protein that loops out from this SH2 domain and places the phosphotyrosine residue from this subunit into the binding pocket of this SH2 domain. And similarly, this one loops out and places its phosphotyrosine residue into the binding pocket of this domain. Now, there's absolutely nothing known about what this domain does in stat biology other than sort of pin the subunits together once they get phosphorylated. And so we didn't know whether this was a problem just with length or whether it was a problem with the sequence. And so we took a couple of approaches to try and figure that out. We made single amino acid deletions, or basically add back is what we did. We had the patient who lacked these 10 amino acids that are in red, and we added them back one at a time going both directions, so that if it was sequence, we would be able to figure that out, or if it was length, we'd be able to figure that out. So, and to try and get at the question of sequence, we did alanine scanning mutagenesis across this loop. And then we did all of the studies, and the bottom line is this. It is that this loop will tolerate, it's simply a length issue, is the bottom line. The loop will tolerate deletion of 8 amino acids out of the loop, which shortens it down to this, 12 or 13 amino acids. And if you look at, and align it with all of the other STAT proteins and you look at their loops, well, that's darn close to the shortest loops that are present in STAT5A and in STAT6, I believe, as shown here. So, and interestingly, if you calculate up the length of those amino acids, if you were to just stretch it tight between the two SH2 domains, it's precisely that number of amino acids. So, you know, it was a lot of work to prove that it's just length, but what's interesting is that this patient's mutation, what it does is it makes it so that it gets phosphorylated just fine, but when it goes to make that big conformational change to get into the conformation to bind to DNA, it can't. It can't be, it's not pinned together, so it just flops. Presumably, we think it flops back to its head-to-tail conformation. It never binds DNA, never upregulates transcription. So we learned something about structural biology of the STAT proteins with this patient. And then there was this group of patients. And so just to orient you here, these are patients who have mutations in what we call the transactivation domain. So to orient you, here's the SH2 domain. Here's that loop that I just showed you, the 10 amino acids that were deleted in that patient I just described. Here's the critical phosphotyrosine residue right here. And here are all of these patients who have mutations just downstream of, and these are all point mutations, just downstream of the critical phosphotyrosine residue. And it turns out that when we began to when we studied these patients, the one thing that unified these patients is that they do not get, when they're activated, the tyrosine just never gets phosphorylated, or at least not very robustly. And so it turns out that these mutations, we think, actually are involved in a region of the protein that is involved in interaction with the JAK2 kinase. Interestingly, despite all we know about the STAT proteins, there's one paper that sort of gives some hints that this is the domain that might do that, but it's never been really clearly mapped. And when we did this, we made all the patient mutations and studied those, and we did alanine scanning mutagenesis from this phosphotyrosine residue all the way down to this phosphoserine residue, which is at 727. And it turns out that phosphorylation is prohibited all the way to 714. And once you mutate past 714, phosphorylation is restored. So all these mutations down here pass from 715 on down. All of these mutants were phosphorylated normally. All of these mutants were not phosphorylated. So it really maps. What's interesting is that, again, the patients tell us the answer. Their mutations literally mapped. You notice that the last mutation is 714. They map the interaction domain with JAK2 beautifully. Now, the one thing that we've not been able to do is to use this region to try and co-IP or to bring down JAK2. We think that this interaction is so transient, we're just having a lot of difficulty confirming that in fact this is the domain. But all of the data suggests that in fact that is the case, because this does not get phosphorylated. And this is just showing that by two ways. So here's the wild type, here's the mutants up through 714. Here we were worried that maybe the phosphostat antibody that binds to the phosphorylated tyrosine residue maybe wouldn't bind because some of these mutations were close to that locus. So we did immunoprecipitations and used a 4G10 antibody that just binds phosphotyrosine and confirmed that in fact that was the case. So again, the patient mutations map the important domain, which is that's the great thing about studying these patients and letting them really guide the science, which is really cool. And that is borne out in the DNA binding. Those that don't get phosphorylated don't bind DNA well. So, what have I told you? So we reviewed STAT biology. We talked about patients who have STAT1 loss of function and gain of function mutations and how they're different. Patients with STAT2 loss of function, STAT3 loss of function and gain of function, STAT4 loss of function, STAT5B loss of function, and some of the work that we've done around trying to understand the mechanisms of these defects and how the natural biology might be able to teach us something about basic stat biology, which hopefully will provide us some insights as how we might be able to drug some of these pathways a little bit more effectively, which would be really one of the ultimate goals of this sort of research. So with that, I just want to acknowledge those who've done all this work, the folks in my lab, Stephen Gulbu for their collaboration on the STAT1 gain of function project, STAT3 project, the folks at our institution, and Ellen and Bernd in Germany, and then on the STAT4 project, our collaborators in Brazil. So with that, I'll stop and be happy to take any questions. Yes.
Thank you so much. It's fantastic. So it's as if you're using patients to do mutagenesis for you then, this last domain of the STAT3 case. So one of the things, at least in the type equivalent stuff that we've done, is we find that there's a big difference in stability of the protein depending on the nature of that missense mutation.
Mm-hmm.
Can that explain any of this at all, or are they basically pretty stable? But is it really a functional difference, or is there more in there?
So the data, I didn't show you the data on that. So they all seem to be pretty stable. There's one that may not be terribly stable, but we think that they're stable because, again, because we don't have any patients who've had deletion mutations, we think that they have to be stable to see the phenotype. They have to actually be expressed so that they can dimerize. And so based on all of the expression stuff we've done, we didn't have cells from all of these patients. Some of them, had died, and we were using autopsy specimens and all sorts of things. So on the patients we were able to look at it, they seem pretty stable. But again, there you've got two alleles and it's a little more tricky. In the transfection experiments, there does not seem to be any significant difference in stability for all but one of them.
I guess on the penultimate slide, when you showed the DNA bind, you're talking about the activation domain mutants, and then you show the DNA binding the gel shift.
Yes. So, and that was actually, what I didn't explain is that that was actually done out of cytoplasmic extracts. So because again, many of them don't get to the nucleus. So for the DNA binding, we had to do those out of cytoplasmic extracts, not out of nuclear extracts. Or, well, actually what we did is we pooled. We took cytoplasmic and nuclear so we could measure sort of the total cell pool of DNA binding activity. So that's, yeah, and that wasn't, I breezed over that, but that was, since they don't traffic to the nucleus, it's obvious. So that they wouldn't if we were just looking at nuclear protein. Yes, sorry.
So with the patients, when you see an autosomal dominant allele, do you think that in those patients you get a partial activation, a partial function of the STAT, and that if those mutations were actually very dominant and completely knocked out the STAT signaling, would that cause embryonic lethality?
Yeah. Yeah, we know, so at least in mice, the only one that's embryonically lethal if you knock it out is STAT3. So the others are not embryonic lethals typically. The STAT5s, you have to knock them both out to get embryonic lethality. But with STAT3, that's absolutely the case. We think that if these were complete knockouts, that they wouldn't be viable. And so that's why we think that and again, getting back to the point that we've never found a patient, despite finding a whole bunch of patients between our lab and everybody else's labs, there's now probably, more than 100, 150 patients that have been identified with STAT3 mutations, and there's not one patient that has a mutation that abrogates STAT3 expression. So it has to be that you're knocking it down below 50% activity, but not to zero because they somehow are all alive. They managed to survive being born. So it's absolutely fascinating. And just from a basic biology standpoint, to think about, 0 is dead, 50% is enough, which gives you a lot of flexibility, there. But 25% is not quite enough. It's enough to get basal function, but not enough to keep you out of trouble.
And that takes me to the question of, so far you haven't found any mutations in STAT5A or STAT6.
Yeah.
Is it possible that those kind of mutations would be lethal, and therefore you wouldn't see it?
It's a good question. We've tried to sort of scratch our brains and think about, what would those patients look like? You think about what would a loss of function mutation in, for instance, STAT6 look like? Well, they might get parasitic infections, I guess, is what you might think about. Maybe they'd get parasitic infections and wouldn't have allergies, maybe?
I would think they would have autoimmune disease.
Maybe. They might. And my prediction is that we'll find them. I think they're going to be out there. The other question is sort of the flip side of your question. Is it a complete knockout? Or is it just knocked down? Is it just not knocked down far enough that it causes a clinical phenotype if the mutations are there, which would be the other side of the coin. And I think either of those are possibilities. My prediction, though, based on how many of these now have been identified, I don't know about STAT5A. I don't know whether we'll find those, but I'd bet money that before this whole gene mining business is over, we'll find some STAT6s. All right. Thank you very much.